Hi everyone, this is lesson 1.1, the role of analytical chemistry, or the introduction to analytical chemistry. For this lesson, we have the following targets. First, to differentiate quantitative from qualitative analysis. Second, to discuss the basic processes of a typical quantitative analysis. Third, relate the role of analytical chemistry in developing society. So let's start with this. How do you know whether an ore contains gold? And how do you know how much gold is in the same ore? So in this illustration, we can see a gold ore. It's a rare matrix gold specimen from Colorado. And here we also have another picture of an 860 kilogram rock from Toei Gold Mine, which is only able to yield 30 grams of pure gold. So how do we know whether an ore contains gold? Of course, we'd have to conduct tests. We'd have to analyze the sample. And the answer that we're looking for is only a yes or a no. Okay, we're not looking for numerical answers for the first question. And the next question, however, wants to know how much gold is in the same ore. How do we know how much gold is in the same ore? So in order for us to know that, we'd have to carry out again certain chemical analysis, but this time we'll have to check that the answers that we get has a numerical value. So in that sense, analytical chemistry is concerned with the determination of the identity and quantity of an analyte in a given sample. So the analyte is the substance being sought in a sample. It's something that we would like to know about the sample, either whether the analyte is present or absent in the sample, or the specific concentrations or levels or amounts of the analyte in the sample. So for example, in the determination of gold concentration in an ore, so gold is the analyte, while the ore is the sample. So here we have the branches of analytical chemistry. We have two. First is the qualitative analysis, kind of self-explanatory from the terms. So qualitative, meaning it's only concerned with the determination of what substance is present in the sample, whereas quantitative analysis is concerned with the determination of the amount of a certain substance present in the sample. And of course, if you'd like to relate the amount of that substance present in the sample, like when you get the ratio of the amount of the substance over the total amount or mass of the sample, of course, you'll get concentration. So next, typical chemical analysis is made up of several steps. So we will start with the first one. First, we choose a method. So in choosing a method, we need to consider the accuracy of the method that we would like, depending on the specific purpose that we want. Do we want to measure the quantities of the analytes in a very accurate manner, or do we just want to check whether it's present or absent in the sample? Do we need specialized instruments? How fast do we want the analysis to be? Of course, the cost, number, nature, and complexity of samples to be analyzed, among others. So in this when we choose a method, all of these we'll have to consider, because we do not want to waste excess time and resources for something that does not really require that much cost or that much accuracy, for example. Let's say, okay, so you can either choose methods involving wet lab or instruments. It would all depend on the purpose of the chemical analysis. So for example, if you just want to check the pH of water for maybe for everyday domestic purpose, there is really no need to get a pH meter that could measure up to 2 decimal places of the pH. If you just like to know if you can use it to water plants, for example, you can just maybe use a litmus paper or a pH paper. Next is acquiring the sample. So acquiring the sample means obtaining a representative sample for a chemical analysis. So the key words here is representative sample. So when we do sampling, we do not just get some stuff just for convenience. Like, for example, you can just go get a bottle of water and a bottle and collect water from a stream, something like that. Of course, there are specific methods that you need to do to make sure that the sample that you're getting actually represents the whole of the population that you would like to describe. Also, this illustration here shows soil sampling. So when you do sampling, like what you have been doing in research, you do not just go where the wind takes you or where it's just convenient for you. So there should be a certain scheme where you'd have to get a sample that would best represent the whole of the population that you would like to describe. There, that's another example. Okay, third step is to process the sample. So this may involve physical processes such as filtration, grinding, and homogenization of samples. So for example, if you'd like to get the pH of soil, so it's not possible for you to just get like the probe of a pH meter and get it in dry soil. So you'd have to process the sample, have it undergo some chemical treatment so that the final form would be in solution. And of course, you can get your readings without harming the instrument. And after acquiring the readings, then you would have to relate it back to the quantity of the original sample. So here's another example. That's a solution inside a cuvette, which will have to be placed there inside a spectrophotometer. And there, just illustrations of how samples are processed before they are actually measured or analyzed. Fourth is eliminating interferences. So interferences are chemicals that may interfere with the detection of analytes in the sample. So this procedure is very specific for every interference. So this is just an example in order to give you an idea of what interferences are. So let's say, for example, you'd like to determine the quantity of, in this case, copper by burning it. So that's in general the principle of atomic absorption spectroscopy. So let's say you'd like to determine the levels of copper in a solution by quantifying or relating the intensity of that green color in the flame. So anyway, if there are other interferences in the solution, then the flame would show different colors, and therefore it's going to interfere with the readings, etc. So if you would like to have an accurate reading for the different things that you'd have to measure, so you'd have to eliminate all of the disturbances or interferences in your original sample before it's clean enough so that the instrument is able to read only the substance that you're interested of, which is your analyte. Next, fifth is calibrating and measuring concentration. So the concentration or presence of an analyte in a sample may be correlated with the response of the instrument or analytical method used. Okay, so this is the actual measurement step already. So notice that it doesn't just jump directly into the measuring concentration part. First, we have calibration. Now, what is calibration? Okay, the calibration step is like teaching the instrument to relate a reading to a corresponding response or value. For example, we have a pH meter in the illustration. Notice that we have three solutions there. One is pink, blue, and the next is yellow. So most of the time, these are called standard buffer solutions. So usually one of the solutions has a pH of 4.0, the other is 7.0, and the next is 10.0. So these standard solutions have constant pH levels over a period of time. So before every analysis or before measuring the pH of a sample, what we do is first to calibrate the instrument. That is by dipping the probe into these standard solutions and assigning values in a way so that the instrument is reminded that this solution is supposed to be a pH 4. Therefore, and the next solution is supposed to be a pH 7, and the final solution is supposed to be a pH 10, so that when it measures the pH of a sample, it is more accurate because it has been like recently refreshed of its memory that these solutions are supposed to have these different pH levels. So if you forget, for example, to calibrate your instrument, then its reading might vary slightly. That's because of a lot of factors, like basically instrument wear and tear over time, and also some inconsistencies with the power source. Okay, next, 6 is calculating the results. So in this step, the analyte concentration is computed whether manually or with the aid of computers from the experimental data. 7 is evaluating results by estimating reliability. So after calculating the results, probably in your previous science subjects, you only get the average and then report it as the final result. However, of course, in higher chemistry, specifically in analytical chemistry, you'd have to estimate the reliability of your results. So if we were able to like do experiments in the lab, then you'd be required to perform at least 3 replicates per analysis, so that you'd be able to get the mean, compute for the standard deviation, and check the reliability of your data. Are they precise, for example? If they're not, then you'd have to make statistical decisions. Okay, so those are the steps in a typical chemical analysis. So here in the illustration, we have, as you can see, these are different colored solutions. Notice that from left to right, their color intensity decreases, and we would assume that whatever colored analyte you have there, its concentration decreases in the solutions from left to right. So um, this is typical of a UV-Vis spectrophotometric analysis. So we would measure the absorbances of light of these 5 solutions, and we would be able to process the data in a calibration curve where absorbance increases as concentration increases. And we're expecting to get a straight line. And from there, we can measure the absorbance of one sample, one or more samples, and then we're going to plot the point or corresponding to the absorbance of that sample so that we can calculate for its concentration. Okay, so that's like in math where you have the equation of a straight line, and then you have one point given absorbance, you have to look for the corresponding x value to determine its concentration. So something like that. There, so that's the end of the lesson. For the navigate part, you already have a copy of this. The research journal entitled Sweeping Micellar Electrokinetic Chromatography for the Simultaneous Analysis of Tricyclic Antidepressant and Blocker Drugs in Wastewater. So you'd have to answer these questions. What problems are being addressed by the research? What analytical instrument was mainly used in the research, and how did it solve the main research problem? And to note, in what other areas can analytical chemistry be applied to address issues in society? So I hope that these illustrations can give you hints of the different areas that analytical chemistry can be applied to address some issues in society. Okay, that is all for this lesson. Bye.